Everything else, let's go ahead and get started. So we have already finished chapter 8, right? And about 50% of your exam is from 8. So anything from chapter 8, we can go ahead and get started with 8. So you want me to do the whole chapter 8 again?
I'm not trying to be funny, but what about our quiz? Is that like 50% chapter 8 too?
I'm not sure. Yeah. It's ready, but I honestly don't remember. If you are not ready for the quiz, then unfortunately you are not ready for the exam. Quiz is everything.
Okay.
Yeah, quiz is everything. Okay, how about Opron? Should we start with Opron? Opron model. Is everybody okay with Opron? Inducible and repressible Opron. Want me to go over Opron?
Sure.
Okay. Let's do Opron model. There are at least 2 or 3 questions on Opron on the test. 2 or 3. What's the purpose of the Opron? Opron is a set of DNA that regulates or controls those enzymes / proteins that are not made all the time, right? 2 types of Opron. One is called inducible. Inducible. This is the one that controls or regulates the synthesis of those enzymes that are digestive. Digestive / enzyme. The other type of Opron is called repressible. This is the Opron that regulates the synthesis of those proteins that are constructive or anabolic. Constructive / anabolic. What is similar between the 2 Oprons? Similarity is that both of them, they have 4 areas on their DNA. 4 areas. Regulator site, promoter site, operator site, and structural site. Same here. Regulator, promoter, operator, structural. This is the area right here, structural genes, that has the information for the enzyme or the protein, right here. Right? Info for protein. Info for enzyme. Major difference between the 2. What's the major difference? What is active?
The other placebo is active.
Another similarity Since the similarity, regulator makes repressor, right? Difference is right here. In both operon, regulator makes repressor, and you can say that this repressor is constitutive enzyme, right? Constitutive protein. Constitutive. I didn't use this term in past. Right? What does constitutive mean? Always present. Doesn't matter if it is substrate is present or not, it is always present. It is constitutive here too. Constitutive. The term I used in lecture was, this is what? Inactive. Inactive repressor. Here it is, active repressor. And the function of the active repressor is what? To bind to, no, to bind to the operator. The function of active repressor is, as soon as it is made, it attaches to the operator site. That is the function of the active repressor. Here it is inactive and cannot bind to the operator. Now, actually, by binding to the operator, what is it actually doing? The process of protein synthesis starts with when the enzyme RNA polymerase attaches to the promoter site. Information for the digestive enzyme is where? Right here. So RNA polymerase must be able to go to the structural gene, but active repressor is blocking its passage. Right? RNA polymerase cannot go to the structural genes. No transcription. Transcription, no translation, no digestive enzyme. Why there is no digestive enzyme? Why is there no digestive enzyme made?
Because of the lack of lactose. There's no substrate or no substrate.
Because there is no substrate present. There is no substrate. When there is no substrate, you don't need the digestive enzyme. Now let's put in this inducer or substrate. Let's put in the substrate now. When the substrate is present, substrate or inducer is present, substrate will make the active repressor inactive. How? Binds to the allosteric side of the active repressor, pulls it away, pulls off, active becomes inactive. No blockade for RNA polymerase. RNA polymerase goes all the way to the structure your genes, transcription takes place, translation takes place, digestive enzyme is. Digestive enzyme reacts with the substrate, digestion will take place. Once the digestion is complete, substrate will come off. Inactive repressor will resume its shape. It will become active again. It will go back to the pathway. Make sense? So what should happen now? Question could be, in order for the digestive enzyme to be made, what should be present? Now for anabolic protein to be made, in order for anabolic protein to be made, what should happen? In order for anabolic or constructed protein to be made, what should happen?
It's always happening. It's always
There is no substrate. In this case, there should be no there is no term substrate. We don't use the term substrate. Repressor must be inactive. And repressor must not be in access. Two things must happen. Did I say substrate? Yes. No, we don't use the term substrate. The end product must not be in access. Two things. Repressor must be inactive, and the end product must not be in access. Two things must happen in order for constructive protein to take place. Let me explain. First of all, repressor protein must be inactive. When it is once it's inactive, RNA polymerase binds to the promoter, can go all the way to the structural genes because the repressor is inactive, right? So whatever you're trying to make, it will be made. Anabolic protein made. So in order for that protein to be made, two things must happen. Inactive repressor, and number two, this protein must not be in access. If you have this protein in access, then what?
It binds to the
Exactly.
and then it stops.
As soon as you have abundance of this protein, what will happen?
It'll stop the process.
Now if I will put this word right here, extra anabolic protein will do what? It will bind to the inactive repressor and make it active. And you know when the repressor is active, it always binds to the and blocks the passage of RNA polymerase. So let me repeat what I just said. In order for anabolic protein to take place, two things must happen. Two things. Number one, what? The repressor must be inactive. And number two, end product must not be in excess or in abundance. If it is in abundance, then it will shut off its own production. Make sense? So that's your compound law. Make sense? What else? Chapter 8. Three processes of DNA transfer. We have already covered two. I'm going to cover all three for you, okay? The last we have not covered, transduction, but I'm going to go over all three for you. Okay? The last one we have skipped, but let's do all three together. There is no eraser here, and I'm not going to mess this up. This is not working. Can someone go to the next classroom and grab an eraser for me, please? Please. I'm just going to use this if I cannot find it. Yes, I've been sitting for 2 semesters. Look, someone has already used it. I've been sitting here for two semesters. Just use this one. DNA transfer. DNA transfer. Between bacteria. Number one, transformation. Number two, conjugation. And number three, transduction, that we have not covered in any of the classes yet. Transduction. Transformation. How many bacteria, metabolically active bacteria, do we need? One. So you have one metabolically active bacterium. It has its own chromosome. This bacterium happens to be tetracycline sensitive. Tetracycline sensitive. It's growing in a test tube. I have to draw a humongous test tube. Okay. A test tube. Here are some fragments of DNA. Naked / free DNA. Free DNA. On this piece of DNA right here, you have streptomycin resistance. This bacterium picks up this piece of DNA right here, it ends up here. Now this bacterium becomes what? Tetracycline resistant. Thank you so much. So, a process in which one metabolically active bacterium picks up a piece of naked DNA or free DNA from its surroundings is called transformation. Okay. Conjugation can take place between two types of male and female bacteria. One, it can take place between male F positive and F negative. You have already learned this in chapter 4. F positive as a pilus makes a copy of the F plasmid, gives it to the female. Both become F negative, the end product is 2F positive. Another type of conjugation that may occur is between a different type of male. And that male, let me draw male first. Sometimes this F plasmid becomes part of the male chromosome. It's not separate, like this. It always attaches itself at the tail end of the chromosome. At the end. Chromosome, but at the tail end of the chromosome. So this is what we did in the lab actually. When we conjugate with HFR and F negative, this is what we did in the lab. HFR, this bacterium is called HFR. High frequency recombinant male. This one is one male. And this is another male. But when this male conjugates with the female, the female remains female. Why? Because the male starts to give this part, the start of the chromosome, to the female, like this. So female will receive some of the chromosome of the male, but she will never receive the tail end of the chromosome. Let me draw it here. This is the male chromosome. Gene number 1, 2, 3, 4, and then 1000 at the end. At the end, then you have this plasmid, F plasmid. Male will never have enough time to give this F plasmid to the female. So she will never receive the tail end of the male chromosome. Why? Because they cannot conjugate for many hours. Because male has to go through binary fission and female has to go through binary fission. The pilus breaks. So female will receive some of the male, but never the tail end. So when these two conjugate, what is the end result? HFR, male, and F negative. Female always remains female, but it's not the original female. She has received some of the male chromosome. And we are going to share the results next time, and I'll prove it to you. What did she receive from the male? When you look at your plates next time in the lab, I'll show it to you next time. We will prove that conjugation does indeed take place in nature. Okay. Just remember this for the test. F positive and F negative conjugate. You have two F positive with HFR and F negative conjugate. They remain same. Transduction.
It's HFR male and then F negative, and then she becomes HFR.
No, she never becomes HFR. She remains female. HFR and female. This is how they started. HFR and F negative, end result is the same. There is a change. Female is not the same, but she's not female. She is not male anymore. She is still female. She has gained some characteristics, but not the pilus. She will not be able to make pilus. Like here, she can make pilus, but here she has not received the red plasmid. She has gained some black DNA, but not the red. Okay. Transduction. How are we doing with recording?
It's going.
What voice? Sound?
I'm tiny.
Sound okay?
Yeah. Yep.
How do you know?
I know. The only thing I can say is there's a little thing flashing on the bottom, but that's it.
Okay, that's a finger okay. For transduction, okay. You need two methods. Short life
She offered it, okay?
is contaminated.
You are something else.
Okay, come back. Now let's demonstrate transduction. Come on, both of you. Right. These two are bacteria. Seriously. These two are bacteria. I'm a virus. Okay? So you need two metabolically active bacteria, okay, and a virus. Virus is called bacteriophage. Phage or phage. I'm going to draw them in a minute. Okay. So why does a virus need to go to a bacteria? So a virus can multiply. Okay.
Parasite has to feed off something.
Exactly. So here's my DNA. Show them your DNAs, please. Color of the DNA is important. So I'm going to go into the first bacterium, and I'm going to start multiplying in this bacterium. After about half an hour, when I come out of this bacteria, I'm not going to be alone. I'm going to have 300 of viruses will come out. 300. Most of my buddies will have red DNA, but some of me, when I leave, I'm sorry, you're dead. So when I come out, I look like a virus, but whose DNA am I carrying?
Easy.
Bacterial DNA. I look like a virus, but am I carrying whose DNA? bacterium. So I go to the second bacterium and give this DNA to the second bacterium. Can I multiply this virus? I look like a virus, but I picked up whose DNA? So I killed myself by transferring bacterial DNA. Exactly. This is called transduction, a process in which a virus mistakenly picks up bacterial DNA and give it to another bacterium. Now this bacteria gain new characteristic, but the virus is gone. Thank you, bacteria. So here is one bacterium. Bacterium A, which is the donor, black DNA, virus, right here, which is called bacteriophage. Bacteriophage. Bacteriophage or phage, which is bacterial virus. Bacterial virus. Inject its DNA into the bacteria. In half an hour, you will have 300 new viruses in this bacterium. I'm just going to draw 3 or 4. Bacteria is going to be full of these viruses. Many of them, they will have original viruses, but one of them, they has whose DNA? Bacterial DNA. Now, if I draw 2 more bacteria here, bacterium C, here and bacterium C here. No. One more thing. The first step is called infection, in which the virus puts its DNA into the bacteria. This is infection. Infection. Process in which the bacterial virus injects its DNA into the bacterium. So now, this virus infects this bacterium, and this bacterium infects this bacterium. Which one is transduction? C or B? C or B? Which one is transduction? Which one is infection?
C is transduction.
This is transduction because this is bacterial DNA. This is infection. This is viral DNA. And that's what is on your review sheet or worksheet, right there, number 8. A process in which a virus transfers its DNA into a bacteria is called transduction. False. A process in which a virus transfers its DNA into a bacteria. No, not its DNA, that is infection. Bacterial DNA. How about it? Yep.
Can you repeat the definition again? You said a
Transduction.
where a virus picks up bacteria from the nation.
And transfer it to a
another
host
bacteria.
into another bacteria.
Bacterium.
Into another bacteria. How about if we make protein now and do some frameshift mutation? Want to do that? Or do you want to do something else? Yes? Yep.
Does this process kill the bacteria?
Oh, this one. This one or this one? This one will.
That will.
Okay. Now, I have not gone into the detail of this. Whenever a virus, now here, I'm showing you that this, I always give the example of Magic Johnson. Is Magic Johnson alive or dead? Whenever a virus infects a cell, bacteria or human, virus has two choices, kill or live. Always. Because we are talking about multiplication, so I'm showing you that it is killing the cell. But virus always has two choices, either to become part of the whole cell or kill the cell. He decides. We don't know. Bacteria may live, bacteria may die. If it lives, we call it lysogenic cycle. If it kills, we call it lytic cycle. We'll talk about that later, so don't worry about it. Virus always has two choices, kill or live. Okay. So let's make protein. Anybody have codon chart? I will bring my codon chart. Book or codon chart? Anybody has book? No problem. You have book? I have a book. Okay. Then I need to, if not, you can go online and pull the chart. Yep. Codons blue, that's going to be really fuzzy. While it's warming up, it's just different form.
I'm so preoccupied with and just do it. And for whatever reason, just take it. And when the other day, you know you should have it. Okay. So I tried to and like one of the codes I'm looking at, it says it's 11, but it's for I haven't said it before, and like, it doesn't make any sense. Just seeing that blue, just like distracted from that. Like, these are just so weird. So I just I don't know. First time I'm doing it. Oh, it's just so weird. Illustrations of it's so that's figured it out.
your DNA, and you are going to make the protein. All set? Go ahead, make protein, please. It's 2 minutes. Stop. You have not asked me which strand is sense strand.
She asked.
I didn't hear her. Where did she ask you?
I did. No, I did ask.
The bottom strand is the sense strand. Bottom strand is the sense strand. So use this, please. So use this one to make your protein. You must start from the start codon. Must start from the start codon, please.
Where's the AUG? Where's the U? Is this the RNA?
I don't know.
Oh, right, THC.
What page is that on? The chart?
Chart?
Yeah, what page is it?
This you have is chapter 8. Chapter 8, page something something. Got it. Chapter 8. Protein synthesis. Ready?
No, wait. Just give me one
Are you
second.
done with the mRNA?
Yeah.
Good. Okay, so I'm going to go ahead and do the mRNA first. I'm missing the letter?
Yes.
Okay, no problem.
You miss it.
What is the next one? L-E-U. L-E-U. A-G, what? Nintendo.
Stop.
How many of you did not get it? No? First sentence. Oh, you all no. It's just DNA is always double stranded, but whenever you make a protein, you always use one of the two strands. Just the sense strand. Whenever you use the N-type, then you replicate DNA. Then you use one strand. I didn't say that.
Yes, you did.
No, making up things. So some of you still didn't get it. Let me do it step by step. Let's practice one more. Yes.
So, like at the end of it, AG was left. You just, there's nothing.
Disregard that. I'm going to do one simple one, step by step. Some of you are still didn't get it. All right. You start with DNA. You have to read the instructions. The instruction said, okay, let's see. C, C, G, A, C, T, A, C, 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 G, A, C, A, T, C. G, G, C, T, G, A, T, So the instructions will tell you which strand to use. So we tell you bottom strand is the sense strand, and you must start from the start codon. Then you just look for TAC in DNA. TAC is the start. So disregard everything else. Look for TAC right there. And TAC in mRNA, mRNA. You know mRNA has 4 nucleotides, A, U, G, and C. RNAs do not have T. So T will bind with A, because it does not have T, will pair up with U. C will pair up with G. G. C, again A with U. C with G. A with U. A and G. Now you have this process, this step is called what? Transcription. The second step in which we are going to use the codon chart is called translation, in which we are going to split these letters in 3, which are our codons. Again, did I say something? Yes. We already know that the first one, AUG, is always MET. GGG is glide. GGC is glide. UGU. C, cysteine, and mine messed up again. Alright. Does it make sense now? Oh, absolutely. What is the anticodon for GGC?
CCG.
CCG. Okay. Now in reality, what is going on? If I remove these, I'm going to put these MET here, MET here, GLY here, and CYS here, and erase these here. Remember, these codons are reacting with anticodon where at A site, U, A, C. Right? This is what's happening. This is tRNA reacting at the A site in the ribosome. This is what's going on. CCG. right? But I'm not asking you to do this. That's what's happening in the ribosome. Yeah. So this is the anticodon. A, C, A. This is tRNA. So if I ask you, what type of RNAs are involved in translation? That's it.
Okay.
tRNA and
mRNA.
This is translation. T. What So, in transcription, what type of RNA are involved?
mRNA.
In transcription, how many types of RNAs are involved? mRNA only. In translation, how many RNAs are involved? mRNA, right? So it's being decoded. mRNA. What else? tRNA. Where is our RNA? So where this is taking place. All three. Yes?
You said that's for translation or transcription?
Translation. That's the transcription only mRNA. Transcription only mRNA. Translation all three. All three.
That's looking at the slide for transcription. It says the synthesis of RNA. It says in parentheses mRNA, tRNA, and rRNA.
Different question.
That's different.
No. Uses, not making. Making and uses. Making of any three could be transcription. Making. The process that uses, the question is, the process of transcription uses how many? Utilizes only one. The transcription makes how many? It could be any one. The process in which transcription, you can make any one of these. But how many it uses? Only one. Okay, one more. Yep. rRNA? Okay, no problem. rRNA. Ribosome. Made up of rRNA. Factory is made up of ribosomal RNA. This whole thing is taking place.
And mRNA is a chain?
mRNA is right here.
Yeah.
That is being decoded at the ribosome.
As it's being made though, just like DNA is a chain, is mRNA a chain?
Yeah.
As it descends.
Yes. All right. Same thing. I'm going to erase this green one, and I'm going to ask you to do a frame shift mutation. Yep.
For a translation, would it go from mRNA to tRNA and then ribosomal, or?
Correct.
Okay.
For translation, first transcription, yeah, mRNA, then to ribosomal RNA,
Okay.
and then tRNA.
But mRNA to rRNA?
mRNA to rRNA, then tRNA.
Okay.
Let's do frame shift mutation.
Do we necessarily have to number in that order, or just remember, translation is all three?
Translation is all three.
it just seems to be. What are you doing?
Yep.
Is different than friendship between?
No, it's an example of example of. Okay. A friendship mutation has taken place at nucleotide number 4 after the start codon. After the start codon at nucleotide position number 4. And 2 guanines are added. After the start codon, a friendship mutation has taken place and 2 new guanines are added. Go ahead, do the friendship mutation and make your new protein, please. Start from the start codon, okay? What position did I say? Number 4. After start codon. After the start codon, let me number them. So I got it all confused. This is the start codon. 1, 2, 3. Add 2 new guanines. Add 2 new guanines and make your new protein.
Live.
Live.
No. Are you changing? Sir, SDR.
You're missing information.
This is what you have to do in the test. Position may be different, but you have to do friendship orientation. It could be adding or deleting. Like thymine is deletion of, thymine dimer is deletion. Yeah.
So like for all the tests, you want us to do the sense strand and then transcript?
Always sense.
Okay. You don't want just the chance.
You want to practice one more?
Wait,
No? Yes?
you lost me.
You're lost? Let's practice one more. I'll practice this. Yep.
We asked if that variation changes the protein.
Of course. Yep. First you have to make normal protein, then you will be asked to add or delete something and then make new.
Never mind.
I think so. I'm sorry, Thompson. I'm scheduled 36 hours this week. I'm really busy. Stuff like that. Well, my only day is off is on Tuesday. My only class is like 11.
Okay, go ahead and make the protein first. This is your sense strand. Oops.
I need to wake up and
This is your sense strand. Make your protein, please. Not yet. First, let's make it normal, then we'll mutate it. Because some of you are still having a little trouble with transcription. Thanks. Okay, now the mutation. At nucleotide position number 5, at nuclear or after nucleotide number 5, after nucleotide number 5, add 2 cytosines, and make your new protein. From the start, ready from this, number your nucleotides from the very beginning. Number your nucleotides from the very beginning, and right after nucleotide number 5, add 2 cytosines. There's only one start. You start right from number 1. That's your nucleotide number 1. And after number 5, add 2 cytosines, please.
Cytosines from the mRNA or?
You don't add anything to mRNA. You always add to DNA.
So we said start after the start codon. You would have start from, you would have said number 1 at G, right?
If that's what the question said.
The only thing that helps me is to the copy of the slides. And go ahead and go through, like, memorize the answers. Just look at the questions, read those questions.
Next.
Wouldn't that be LEU?
It was
CUG, LEU.
LEU. Okay, practice that at home. Okay. What's next? Chapter 8. Anything in chapter 8 before we go to chapter 7?
I have a question. For the DNA replication,
Pardon me?
the DNA replication.
Yes, application.
Can you further explain, like go into detail for like step number 8 and number 7?
What is it? Just
It says RNA fibers are removed and opposite fragments.
we are not going into that detail. Yeah. Just I'm going to give you one strand of DNA, you have to give me another one. We are not going into Okazaki fragments and leading strand and lagging strand. Yep, not going into that detail. Yep. Yes, I'm going to read the copy. Yeah. Chapter 8. I'm going to just go over one more time the cat DNA and dog RNA polymerase. One more question. Okay.
It said E, but there's no E, right? Still or something? I don't know. But it said E. Whatever last name was that, it's just.
So in a test tube, you have mixed DNA from E. coli and RNA polymerase from Entamoeba histolytica. What is the end product? E. coli what? So here is the DNA from E. coli. T A C G G G B C A T C. E. coli DNA, and now you have the same color. So RNA poly from E histo. So we are going to make A U G C C G U A G. So what did we make? RNA. Whose RNA? E. coli RNA. Right? Thank you. Draw it. You are allowed to draw, but half of you said DNA. I heard it on witness. Because RNA, there is RNA, so you make RNA. Nothing. Easy peasy. Chapter 7. That's 6. Yes. No, not necessarily. Anti simply means against. It either kill or stop. May kill, may not kill.
But it will stop.
It will stop. May kill, may not. Yeah. 3 methods to sterilize heat labile material. Okay. Number 1, ionizing radiation. Number 2, pentalization. Number 3, ethylene oxide gas. Good. So, ethylene oxide gas, pentalization, and ionizing radiation. And there is one more. Glutaraldehyde. Glutaraldehyde. Under the headings of aldehydes. Only one. Not formaldehydes or others. Glutaraldehydes. And if you want small quantities of liquids, you can also use 0.01 micrometer filter. Small quantities of liquids. All quantities of liquids can be sterilized by using filter 0.01 micrometer. Yes, for my student.
She had a question.
Yes.
Pentalization, ionizing radiation, glutaraldehyde, and what's the fourth one?
Pentalization.
I have one.
What was the fourth one? You already have that.
Urea aldehydes.
Ethylene oxide gas. All right. Chapter 7. Autoclave is one of the methods that is commonly used to sterilize material that are heat resistant, operated at 121 degrees, 15 pounds per square inch, 141 degrees, average time 20 minutes, could be 10 minutes to 40 minutes, 141 degrees, 15 pounds per square inch. 121, 15, yeah, 121. Which one? Boiling is always disinfectant, never sterilized. What?
Why, boiling is a disinfectant while if you put something in, it'll sterilize when you're doing the.
Pressure cooker will sterilize it because that is steam under pressure. Boiling just has no pressure. So if you want to sterilize something at home, put it under the pressure cooker.
Oh, it's moist, that's what I was thinking.
No, boiling is moiety too,
Yeah,
but there's no pressure.
the pressure is what kills it.
Chapter 7. Yep.
So what happens to bacteria if you put it in high temperature?
Dry heat or moist heat? Only meant or both. Now, depends what type of moist heat. Dry heat, there is only one dry heat that will accomplish sterilization. Not one. Incineration will definitely accomplish sterilization. Dry heat, the disadvantage is it takes too long. 3 hours, 180 minutes, 140 degrees, 3 hours.
And that one, it only uses oil and powder.
Yeah, anything that's hydrophobic.
You didn't really go in.
Moist, 121 degrees, 15 pounds per square inch. All of it. Or sterilization. 3 days.
Which method gives partial sterilization? Which method gives partial sterilization?
My beautiful.
Gives partial sterilization?
Partial sterilization, there are several.
There's supplements?
None. Partial, there is no method that accomplishes partial sterilization.
But there is fraction.
Partial, there is fractional, but there is no partial. That's their nature. Yes, somebody said that I. Chapter 6. Yes.
Okay. So, for Bacanthi agar, it has crystal violet and lactose. But which bacteria favors crystal violet? Because when you ask the question, if crystal violet isn't in the Bacanthi agar, all bacteria will grow. So which bacteria favors crystal violet and favors
Gram negative. The Bacanthi has crystal violet. It stops the growth of gram. Gram positive. So if you remove it from Bacanthi, all will grow, both gram positive and gram negative.
and crystal violet and a gram negative vapor source.
So the crystal violet is taken out. Does that mean it loses its selectivity?
Correct.
So for concave are selective and different.
Chapter 6. Can someone else ask questions too? I'm sorry. I'm sorry. I know, you can. Let's give someone else a chance to please, yeah. I know, yeah, sure. You can do it, but yeah. Yeah, sure, we can ask you, sure. But give everybody else a chance. You can ask questions, for sure.
Like the question
Please. That's a bad question. I'm going to rephrase it. So chop it up.
still go?
Sure, we can go with it. Go ahead.
Okay, so the first one is substantial reliance. Yeah.
What can you do?
Paul, how long will it take place?
Those are equally modified by computer
What's the other option? Number? 18. Microcyclone. Should have choice E to there. They should have choice E. Yeah.
E?
So
A and D should be A and D.
it would cover A, but A
I would remove the word substantial. Yeah.
So it's
And D.
and
E, choice E, and D. Yeah, both, A and D, both are.
So for 18 it's A and D.
A and D.
But what about C?
That's what I'm saying, just take it out. It needs to be rephrased. The whole question needs to be corrected. I'm going to fix it. If I put this on the test, I'm going to fix this. Because there are too many possibilities in this question. I'll take it out. Any other question that may be confusing or which one? 8. 8. Steam under pressure. It should be D. Steam under pressure is associated with sterilization with moist heat. Correct? Temperature of 141 degrees, correct. Autoplay, all of the above, correct. So I think the P says E, but it should be D. It's a typo here. Number 8 should be D, P says E. 65. 65, yeah, we just did that. So what do you think? Answer C. And you should be right. According to the Opron model, for the synthesis of an inducible enzyme to take place. 65. Substrate must bind to the repressor, or you can say active repressor. Active repressor, right? Anything else in here? 27 C. 27 C. Oh, I'm sorry, not C.
D? D you mean?
That correctly. Log phase vectors do not divide, that is correct. They do divide. C that's correct. That's good. Lag phase, B should be lag phase, and the answer should be B. B. Lag, number 27. B. Lag phase. Binary fission, method most vector undergo when enter lag phase, not lab phase. That's the only one that is correct. The other two are incorrect. And the question is, which of the following pairs match correctly? The other two are incorrect. No, it is not incorrect. Bacteria do not require. Yes. Yep.
So 27 is
That was
B and C?
yes.
Both of them.
That is not true. Not true. But they prefer. They prefer, right? It says match correctly. Match correctly. It's not match correctly. Another question before we go to the next. Let's do this last. What's the question of the test on this one? Candidates negative. Candidates negative, accurate, incubated, aerobic. Which line depicts the growth of pathogenic negative bacteria incubated in the presence of oxygen? Catalase negative bacteria incubated in the presence of oxygen. A bacteria that cannot make catalase. When it is grown in the presence of oxygen, what will happen to it? Die. Have no catalase, will die. Cannot neutralize toxic oxygen. See. Catalase negative bacteria grown in the presence of oxygen. What's this next one? Facultative anaerobe grown in the presence of oxygen. Facultative anaerobe grown in the presence of oxygen. A. Preferred to grow in the presence of oxygen. Let me ask you another question. Facultative anaerobe grown in gas path. Facultative anaerobe such as E. coli grown in gas path. They prefer to grow over here, but they can grow right here. Aerobe, such as Micrococcus luteus or Mycobacteria or Legionella, grown in gas bath. Same bacteria grown in thioglycolate.
So all aerobies would have to have catalyst.
McConaughey minus CV. McConaughey minus crystal violet. Staph aureus. Which line shows the growth of Staph aureus or McConaughey without Staph aureus? Okay.
Explain that.
It's gram positive, and if it does not have crystal on it, it will grow beautifully. Medium is still selective or different. Is the medium still selective? No. Is it still differential?
Yes.
Yeah. What will happen? Anaerobic gas. What will happen to bacteria and yeast?
Yes.
Anaerobic. Actually, I'm not Oh, anaerobic bacteria. In the gas. me which line shows the growth of which bacteria in gas packed jar. These three bacteria. Number 1. How many think A? For number 1, Clostridium sporogenes. A. A is correct. In gas packed jar. E. coli. E is correct. Micrococcus luteus. C is correct. Gas packed jar. Because Micrococcus luteus is aerobic. E is facultative anaerobic. And Clostridium sporogene is anaerobic. Most of you missed that question on the midterm because you thought it is obligate anaerobic. It is not obligate anaerobic because it grew in tube glycolate. That's why. Okay. Let it go. I'm not going to go there. Okay, because
We're going to do that one question?
nope, not really. I already did that.
Oh, that's right. You did.
In the book, it says genus Clostridium is obligate anaerobic. But when
Not all of them.
that's the, but when you look at the Burmese manual, it says that range some of them
I don't agree with the book all the time. They make so many mistakes.
send them an email.
Write the book.
Even though it can grow at 100% CO2, that doesn't mean that it can't tolerate
Oh. Staphylococcus epidermidis. Oh, you can.
No.
Oh, I thought. Sorry. Staphylococcus aureus and Staphylococcus epidermidis on mannitol salt agar.
See?
Which line shows the growth of Staph aureus on mannitol salt agar?
B.
And Staph epidermidis?
B. No. A.
Both A. Don't miss it on the test. Salt concentration of MSA is reduced to 0.9%. Salt concentration of MSA is reduced to 0.9%. Which line shows the growth of these two on mannitol salts?
C.
Still A. They are facultative halophile, not obligate halophile. They will still grow the best because they still grow on mannitol and on nutrient agar. So they will still grow the best. Growth of Helicobacterium salinarium on mannitol salt agar.
C.
Growth of Helicobacterium salinarium on mannitol salt agar.
C.
They're off.
Yes.
Mannitol salt has 7.5%, it needs 12%. All right.
On the pretest where it says, okay, Okay, for medium B is at 12%. That would be an unknown composition, right?
One more time.
It says 20% salt. That would be unknown composition, right?
That would not be unknown concentration, but that is unknown salt. Concentration is known, but the salt, what salt? There are so many different types of salt. So that is unknown. Chapter 6. Oh, so you're asking her now.
Mental salt, is it selective and differential?
Mental salt? It is. Chapter 6. Going once? Yeah. Yes?
Okay, for the Calvin-Tempel/Dock reaction, what bacteria is the Dock reaction or Calvin-Tempel?
That's chapter 5. Okay, we are moving on to chapter 5. Chapter 5. That's chapter 6. If you can read the chemical composition, I think everything is spelled out. Carbon, hydrogen, nitrogen, phosphorus, that is chemically defined. If you just say salt or amino acid or protein, it's chemically complex. Oh, sure. Yes. Well, Bergey's manual. Bergey's manual. B-E-R-G-Y-S. Bergey's manual. But after looking at that, you probably want to come back to my notes.
Bacteria that heat loving?
Thermophiles. Bacillus stereothermophilus, Bacillus, Streptococcus thermophilus. I think I gave you 2 names. Tasha?
Transport, diffusion, that's the kind of one.
Diffusion? That is fine, yeah. No, that's 6. No, that's 6. That is 6. You want me to go over there? Sure. How do bacteria transfer nutrients across the membrane? three methods. Fourth one they cannot use, which is phagocytosis. Yep. Phagocytosis, or pleocytosis, they cannot use because they have cell wall. Diffusion, simply movement of molecules from high concentration to low concentration. Osmosis, only water molecules. Facilitated diffusion through channels. Yeah. Amino acid, glucose molecules, they move from higher to lower, but through carrier proteins. Active transport and group translocation, same thing. But movement of molecules from low to high. Carrier proteins are involved and ATP is involved. The only difference is molecule that is being transported remains the same here. Over there it is. Why is it altered? The cell needs it immediately. Glucose go in, remains glucose. Carrier protein, ATP is required. Transported molecule remains same. In this process, group translocation, molecules goes in, ATP used, but the transported molecule is chemically altered. The example that I gave you was what? This is glucose, and this is what? Phosphate. This is the first step of glycolysis. This glucose will enter into glucose at step number 1, and this glucose will enter into glycolysis at step number 2. So when the cell needs to make glucose faster, this is what it does. Group translocation. Chapter 5.
We have to know the specifics of aerobic respiration, like glucose 6-phosphate turns into fructose 6-phosphate.
Do you also know the specific organisms that are used during fermentation?
One more time.
Do you want us to know all the microorganisms?
No, just the definition. Just the definition. The broad concept.
For fermentation?
Yeah. I may give you the organism as long as you know what fermentation is. That's it.
Okay.
How many ATPs are made? That's all.
In class on the slide, it says for Krebs cycle, it's 6 NPHs, but then on the topic, the list topic, you said 8 NPHs.
2 actually come from the transition reaction. 6 are made in Krebs cycle, 2 from the transition reaction. From formic acid to acetyl-CoA. Separate, exactly. Chapter 5, the end products of everything. This here. And carbon is attached to IUPAC. Yes. So, feedback inhibition. What's the difference or the similarity between feedback inhibition and enzyme repression? Feedback inhibition. This is a process by which cells, they regulate or they control the synthesis of end product. When the end product is plentiful, it's not needed, the end product will shut off its own synthesis. Same thing happens in enzyme repression. This is the operon model. Operon or repressible operon. Repressible operon. Repressible operon takes place at the DNA level. Both are similar. In feedback inhibition, you start with an enzyme. The example I gave you was a capsule. Bacterium is trying to synthesize a capsule. It has synthesized its capsule. You need 1, 2, 3, 4, 5, 6, 7 million molecules of this amino acid to synthesize this capsule. And this is the metabolic pathway that makes these amino acids. Right here. This is the end product right here. End product. Then you have extra end product. Extra end product. Extra end product is going to go up, bind with the allosteric site, and change the active site, and shut off its own synthesis. And that's called feedback inhibition or end product inhibition. And product inhibition. In enzyme repression, same thing. when you want to make a product, anabolic protein, regulator gene makes a repressor protein, which is what? Inactive. By being inactive, it cannot bind to the operator. When it is inactive, RNA polymerase can travel to the structural genes because this cannot bind to operator. So transcription takes place, translation takes place, and anabolic or constructive protein is made. When you have excess or extra anabolic protein, extra anabolic protein binds with this inactive and makes it active. An active repressor always binds to the operator and blocks the passage of RNA polymerase and shuts off its own synthesis. So that's how the two processes are similar. End product inhibition, same as anabolic repression. Non-competitive inhibition, you remember, non-competitive inhibition. No competition between the substrate. What binds with the allosteric site? Heavy metals. Heavy metals. Substance that is not made by the cell itself. It comes from outside. Heavy metals. They bind to the allosteric site. This is irreversible. Those are reversible. This is irreversible. Changes allosteric site. No end product. Comparative inhibition. We have two molecules that compete for the same active site. Allosteric site does not play any role. Gave you two examples in lecture. Kava is normal. Kava. I gave you example of sulfa and AZT. Yep. If the normal substrate goes in, end product made. If lookalike goes in, no end product. This is called molecular mimicry. That's called. Or comparative. These lookalike, they compete for the same active site. Mimicry. They mimic the original molecule. Now, initially when it is made, it is always inactive. Made becomes and blocks its own synthesis. What else? thymine. AZP is an analog of thymine. AZT, which is the HIV drug. Yes, go ahead.
My question for chapter 8. On the worksheet, you said provide the nucleotide sequence cell code for the following amino acids. And then 4A says LE, TRP, HIS, ARG, and GLB. How would I go about that to answer the question?
Chapter 8. So we are going to open the form for all chapters. So you can pick any of the codons. So there are more than one codon for each. So you can pick any of the codons, or there are 6 of them for LEU. You can pick any of those. So I'm going to pick one example that you said. So let's do that one. Read me the first 2 or 3, please. LEU, TRP, and HIS. So LEU has 6 amino acids. So I think the results or the answer that I have given you. What question number is this?
4A.
4A.
I want to ask you a question.
One second. All right. Now these codons that you have right here, I have only picked the very first one in the codon chart. You can pick any one of them and the answer will be correct. Right here. So this one has 6. I'm not sure. Tryptophan only has 1. You cannot go wrong with this one. This one has, I think, more than 2 or 3.
It's
So,
going backwards though?
pardon? Backwards. So this is mRNA, this is tRNA.
What number was that?
Yes. I honestly know.
Yes, Monday, Wednesday class. I was the person who said it, so I know. And I don't have class on Saturdays. Sure. We agreed on this.
But this is on the test.
Take it off. Put something in
This is the important thing you have to know. You have to know this. Yes, you do.
So, okay, so the answer would any.
Any amino acids.
So how are you gonna answer? You're gonna
Oh,
have.
believe me, very nicely. You'll be able to do it.
So just as long as.
Right here. You have the. Just pick any. Okay. TRP, C. Okay. LE. Yeah. Okay.
ASM.
Okay.
ALY.
ELY.
AST.
Okay. Yes, but that's enough. That's enough. All right. Go ahead. Take the codon chart. All you have to do is look at the codon chart. All right. Codon chart and pick up the first amino acid, ASM. exactly. ASN, let's say then CCU. DLY is AAC, ASN, CCC. That's it. These are the codons. And then we have to go one step back to make, first to make your mRNA, then you make your DNA. G, A, T, G, G.
So we have to find the DNA too?
Oh, DNA 1. Just kidding.
So wait, but then how, it's gonna take forever, no?
Why?
Because then we have to go,
Okay. Normally I give you DNA and you make protein.
Right.
What's the big deal if I give you protein and you make DNA?
There's so many different,
If you give them the
Okay. Look at the example that I have in my pretest.
yes.
See, if you do that, you're good. Yeah, I have the same example in my pretest. That tells me no one has done that yet. That tells me that you're way behind.
No.
we're studying first.
Alright. So if you have done that example, you're in good. Let me tell you, same example is on the test.
There's how many questions?
That's an example? Oh no.
Okay, what's the one? Same example.
I'm not supposed to say that, but I already said
Okay, so are you going to memorize? The first one, you that mRNA?
yes.
Yeah.
Why is that mRNA?
I think that's
Because it has you.
Because of the use.
Oh, yeah, that's all right. Thank you. You're not going to go from that and then tell me the translation, right?
Yes, I will do that.
You do that too?
Yes.
But how do we know for guessing?
You're not guessing, everything will be very precise, don't worry. Any other questions? Yes. Irene? Yes.
Like, but all the ones are in that. And what's in that transfer
What time is it?
2:18, yeah.
Any other questions? Good luck. Enjoy the rest of your week.
Enjoy.